So when they asked for a bio, I never, ever expected anybody to go ahead and read the whole thing out loud. But I do want to thank the Illinois AFS and the Illinois Lake Management Association for inviting me to give this talk. It's a really great honor to be here and to do this. And actually, I can skip over some of my introduction because of what you just did. So, as we just heard, the core of what I'm going to be talking to you about has been supported by the long-term resource monitoring element of the Upper Mississippi River Restoration Program. I've also got some information from an NSF research experience for undergraduates project that we had last year. I was a co-PI on that, and the PI was Carol Colomino Meeks, who is now at Southern Illinois University, Edwardsville. And then the long-term survey and assessment of large river fishes in Illinois is better known as the LTEF project. That has been going on in various forms for 50 years, recently expanded onto other rivers besides the Illinois River. And my colleagues, Andy Casper, John Epifanio, and Young Sao at the University of Illinois are PIs and co-PIs with me on that. And I wanted to mention that Daniel Gibson Reinemer is a postdoc that Andy and I share, who has contributed a lot to some of the information I'm going to be talking about. All right, so let me explain a little bit about the long-term resource monitoring program. It is a system-wide program monitoring fishes, water quality, and aquatic vegetation from 6 reaches of the Upper Mississippi River system, starting in Pool 4 in Minnesota, including Pool 8 in La Crosse, Pool 13 in Iowa. Then we also have the LaGrange Reach of the Illinois River. The best reach, though, is Pool 26 near Alton, where I am located and all my crew work. And then finally, we also have a reach in the Open River, just above the confluence with the Ohio River near Cape Girardeau, Missouri. At all of these different locations, we have the same monitoring design. It's a highly standardized monitoring design. For fisheries, we have a multi-gear approach and a multi-habitat strata approach. So we sample the main channel, side channels, backwater lakes, and our design allows us to basically calculate a pool-wide mean for each year that cuts across all of the different habitats. So a little bit of background on Asian carp in the Upper Mississippi River system. Of course, the very first one of these invasive carp to come along was the common carp, which even some people to this day think is a native species because it was introduced back in the 1870s, very early on, and but is in fact not native to the United States and is an invasive species. The next one really to come along and become established was the grass carp, followed by the silver carp and the bighead carp. And today, unfortunately, I can report that we can officially welcome black carp as reproducing Asian carp in this system. Our colleagues down at the Open River Field Station near Cape Girardeau this year captured several young of the year black carp in their monitoring, and so that's real evidence that they are indeed reproducing in the Upper Mississippi River now. But let me focus a little bit on grass carp, silver carp, and bighead carp, our kind of most recent invaders besides the black carp. So this is some work by Mike Wilson, a former graduate student at the University of Illinois. He looked at records to calculate frequency of occurrence of these 3 species. He used LTRM, LTEF, also IDNR monitoring data, and the Illinois Natural History Survey collections. And basically, black carp came along, grass carp came along in 1971. The next record in Illinois was silver carp in 1983, and then bighead carp in 1986. So very different dates of introduction. Obviously, different lag times, the longest being grass carp. But then when you get to about 1998, they all go through this explosive population and geographic expansion at the same time. And we still don't know really exactly what kicked them all off at that time. Now, for the rest of my talk, really the only carp I'm going to be talking about are silver carp. And so, continuing this review, I wanted to just kind of go over why these silver carp are doing so well in the lower portion of the upper Mississippi River system. So what do silver carp need to thrive? Well, their spawning requirements are they need water temperature to be at 18 degrees C or higher. Once it reaches that temperature, they're ready to spawn. The next thing they need is a rise in the water level. That kicks off their behavior. They release their eggs into the water column and they do their thing. Once the eggs are produced in the water column, they need velocities from 0.3 to 3 meters per second to keep the eggs in the water column until the larvae are ready and able to do that themselves. They also need they need food, they need forage to consume. By and large, silver carp feed on phytoplankton and zooplankton. When those resources are scarce, they will shift over to detritus. All right, so if we look at the conditions in the lower portion of the upper Mississippi River system, we can see that temperature requirement really is not a problem. These are fixed site water quality data from the LTRM from Pool 26 and the LaGrange Reach of the Illinois River. I restricted these data to April 1st through October 31st because in the winter months, some of the collection of the data can be a little bit sketchy due to weather conditions and ice or lack of ice on the river. So this is a much more complete and consistent data set. And basically the take-home message is we've got significant increasing trends both in Pool 26 and the LaGrange Reach. A little more on that later, but clearly there's plenty of opportunity for water to be above 18 degrees centigrade. Then the whole upper Mississippi River system is really a large flood pulse, large floodplain river. And so the rise in water level also really isn't an issue. Now in Pool 26 on the Mississippi River, I'm going the wrong way. Before the construction of the lock and dams, you had a very predictable spring flood, pretty large magnitude. It's still there. It's just the magnitude is reduced because we don't let the river go to a low level because the whole idea of these lock and dams is to allow commercial navigation to pass. But still plenty of opportunity for a river rise and for them to spawn as a result. Moving over to the Illinois River, pre-construction of lock and dams, again, a very consistent spring flood pulse. But the lock and dams on the Illinois River work a little differently than those on the Mississippi. These dams, the gates lay flat on the bottom of the water, and when they need to raise the water level, they'll lift up those dams a little bit to bring the water level up, and then they'll lower them. And since the Illinois River is a smaller river, it responds to water inputs a little faster, and then they're always adjusting those gates to try to get the water level right. And the result is, today, we have a system where there's lots of smaller rises and fall of the water level. Not all of those rises are going to be major floods, but it's much more flashy in the Illinois River, and that may be a reason why they do so well in that river. All right, so what about their forage base? And to get at this a little bit, I'm going to show you some data from the Great Rivers Ecological Observatory Network, which the National Great Rivers Research and Education Center supports. We are involved with National Great Rivers Research and Education Center. In fact, that's where we're housed. What we're doing with that program is putting out these real-time automated water quality monitoring platforms throughout the upper Mississippi River and also in some areas of the watershed. So I'm going to show you some data from Pool 8 in Wisconsin and Pool 26 where I am. And in both of these cases, we had a paired platform, one in the main channel of the river and 1 in a backwater lake. These data are the concentration of chlorophyll A in the water column, so that's the abundance of phytoplankton in the water column. And you can see in pool 8, chlorophyll A concentration was always greater. These are data from 2015 in the main channel than it was in the backwater lake. And that's because up in pool 8, backwater lakes are filled with aquatic vegetation. They're known to be kind of sport fish paradises. On the other hand, when you look at pool 26, the pattern is exactly opposite. Here's chlorophyll A in the backwater lake. It's always higher than what we see in the main channel, even though there's quite a bit of chlorophyll A in the main channel as well. But here in pool 26 and in the whole lower portion of the UMRS, the suspended sediment load is way too high for submersed aquatic vegetation to grow anymore in our backwater lakes. So we have no submersed aquatic vegetation. All that primary production gets shifted to phytoplankton. So in pool 8, we've got lots of aquatic veg, and we have a sport fish paradise in those backwater lakes. And down here, we've got no aquatic vegetation, and we've got an Asian carp paradise with all that phytoplankton. So we've got good spawning, plenty of forage. Silver carp should be able to thrive, and indeed they do thrive. I'm showing you all the silver carp we capture in each year from all of the gears we used and all of the habitats that we sample. And these are for, again, the LaGrange Reach of the Illinois River, Pool 26, and the Open River. I want to point out that these data are plotted on a log scale. So keep in mind, as you compare the red bars, which are the LaGrange Reach of the Illinois River, to the blue and the white bars, that's an order of magnitude difference. So that's a big difference going on. LaGrange, in a year, they catch close to 100,000 at their very peaks, whereas the peaks at Pool 26 and the Open River are only about 5,000 in a given year. To give you a little better feel for what an exponential increase in population size is, I've got just a couple of videos to show you. The first one is from pretty early on in the invasion of these things in Pool 26. That's Eric Ratcliffe and Eric Gittinger from my crew out on the river. At the time we filmed this, you would hear the voices going, "Wow, this is really exciting," and that's what we were trying to do, capture good video of these things jumping, which was pretty amazing back then. That was shot in 2004, in the kind of early days of this invasion. Now I'm going to show you a video from 2010, a little later in the invasion. And this will give you a feel for what an exponential increase is like. So this is again in Pool 26. So now I'm not going to claim that this is a scientific comparison, but in both cases we're out trying to get the best footage we can. And this is what you can run into today, whereas we would never have experienced that back in 2004. Some more information before we move into some other portions of my talk. I'm going to look at, again, using all the data from LTRM for the LaGrange Pool, Pool 26, and the Open River Reach to show you how we can track or really see year class strength with our data. So again, these are all the fish captured from all the gear and all the strata, but what I'm plotting is each dot is either a fish or a group of fish at the same length. So you can see the first strong year class in LaGrange was produced in 2000. And you can see that year class follow on through. So a very strong year class. Then again in 2003 and 2004, you had strong year classes produced. Then again in 2007 and 2008, really strong year classes produced. Then 2014, another strong year class. We'll see if it recruits into other years. So these data are really unique in our ability to just have so much of it to be able to do that kind of a demonstration. So now let's go downstream and look at Pool 26, and let's see how similar it is to this pattern we see in LaGrange. And you can see that Pool 26, in the early years, we did not have that really huge, strong year class in 2000. Now, we noticed reproduction, and it was a pretty strong reproductive event for us, but not nearly as strong as in LaGrange. And then finally, when you get to 2007, you see a year class formed, 2008, 2009, 2010, 4 years in a row. That's very different than we saw in LaGrange. And that corresponds to really 4 or 5 years in a row of some very strong flooding. I'm graphing the number of days the river was in flood stage, and you can see 2007, 2008, 2009, 2010, even 2011, although for whatever reason we didn't get a strong year class that year. But major flooding really does promote the spawning of these silver carp. If we go even further downstream to the open river reach, now this is really different. Really, one thing I'll point out, if you look at the fishes 100 millimeters in length or less, there's consistent reproduction going on. You know, they had good reproduction in 2000, 03, 04, 05. But really, in this early time period, there was no recruitment from this reproduction. They did get spawning and recruitment in 2008, good spawn in 10 with a little bit of recruitment, but compared to the other reaches, you're seeing lots of reproduction, but reduced recruitment in the open river reach. And unlike Pool 26 or the LaGrange reach, 95% of the floodplain in that open river reach has been permanently levied off, and there are no backwater lakes. So that lack of backwater lake and floodplain habitat may very well be limiting recruitment down in the open river reach. There could also be emigration out into more favorable areas of the river. So to summarize this introductory material, after different lag periods, those 3 Asian carp started their big geographic and population increases really at the same time, between 1998 and 2000. The lower UMRS certainly provides favorable water temperature and water fluctuations for silver carp reproduction. We've got reproductive success evident in the entire lower UMRS, but recruitment in the middle Mississippi River is limited, maybe by reduced floodplain or backwater habitat. Also, probably there's some emigration out of that area going on. But by and large, silver carp populations are increasing exponentially in the lower upper Mississippi River system. Now I'm going to shift a little bit. Some of you in AFS have seen some of this information before, but I'm going to go into greater detail to bore you guys to death, hopefully. We're going to talk about silver carp and how they interact with native fishes. We know from a lot of research that's been done in this system already, a lot of it done on the Illinois River, there's dietary overlap between silver carp and our native filter feeders, namely gizzard shad and bigmouth buffalo. Work done by Kevin Irons and company on the Illinois River shows that, to go along with that, condition factor and the overall abundance of gizzard shad and bigmouth buffalo have been declining, and those declines kind of correspond with the establishment of silver carp. Greg Sass has shown that the zooplankton community in the Illinois River varies, and their community structure variation corresponds with Asian carp abundance in the Illinois River. And community structure and composition of native fishes. This is new research from Levi Solomon and company, including myself, that is really in press right now. And in any case, the community structure of native fishes shifts after silver carp come into this system. So a lot of information that things are happening, most of it from the Illinois River. What I wanted to do is look across the whole system and take advantage of all the data that the LTRM program has. And so when we look, we've got these 6 reaches. We've got the 3 reaches up north, Pool 4, Pool 8, and Pool 13, and silver carp have not really established populations there yet. So we've got 3 reaches that can be controls. We've got 3 reaches, LaGrange, Pool 26, and the open river, that are impacted by silver carp. Here's the abundance of silver carp. This is catch per unit effort from electrofishing at these 3 reaches. So we're really in an excellent shape to look at a natural experiment and to analyze the data to take advantage of all of this. We've got 3 control reaches, 3 impacted reaches. We've got 20 years of repeated measures from each of these reaches. We have 6 years before the establishment of silver carp and about 14 years after the establishment of silver carp. This is primed for kind of a before-after control impact analysis, a BACI analysis. Wanted to do this using the pool-wide means for electrofishing, and the indicator I wanted to look at was the combined abundance of sport fishes in these reaches, which immediately begs the question probably in a lot of your heads, why sport fishes? I just talked about our filter feeding fishes. Well, for several reasons. When we think regionally, I mean, we're going all the way from Minnesota down to Cape Girardeau, Missouri. There's relatively consistent regional management goals for sport fish to either maintain or increase their abundance. Additionally, most of our sampling gear used in LTRM are known to work well for sport fishes. In many cases, we're designed with sport fishes in mind. Arguably, you could say our monitoring design is also biased towards sport fishes. We do a lot of work on the shoreline in all of these habitats. Regardless of the bias, lack or not, the trend data for sport fishes should be as good or better than any other indicator group that I could pick. Then when you think about the definition of an invasive species, a species causing ecological and/or economic impact, if they're causing a negative impact on sport fishes, that's both a negative ecological impact and a negative economic impact. So that's why I wanted to look at sport fishes. Now, the traditional way to analyze a BACI design is to use a simple analysis of variance with 2 classification variables, the treatment, control and impact, and the period before and after. And you run the analysis and you look for a significant interaction. And indeed, I can do that, and it is significant. There is a significant interaction. Regardless, I don't think that's the best way to be analyzing these data. I really think we need to be taking advantage of all these repeated measures we have in all 6 of these reaches. Additionally, BACI treats that impact as a single point in time impact that happened and then has been the same ever since. Well, we know that silver carp vary from reach to reach where they're present, and they've been increasing throughout time. So we wanted to take advantage of all that and to also challenge the relationship between silver carp and sport fishes by introducing other variables that also differ between the control reaches and the impacted reaches. And so for those, I'm choosing water temperature, suspended solids, and flooding. So, as you might expect, the water temperature in the 3 lower reaches is much warmer than in the 3 upper reaches. So it nicely separates control and impacted reaches. Good way to challenge that relationship. What's a little bit disturbing to me is there's a significant increase in temperature in the main channel of this river at all of these reaches. That increase is remarkably parallel at all 6 of those reaches. Over on the right-hand side is total suspended solids in the water column. And again, it's much higher in the lower reaches, much lower in the upper reaches. Here, there's a little more variation in the trends from reach to reach. Finally, we looked at the number of days in a year when the river was in flood stage. We've got the upper 3 reaches on the left, the lower 3 reaches on the right. I think everyone can see flooding is much more severe in the lower reaches. In fact, it's much more severe in Pool 26 in the open river than it is on the Illinois River. Not to say that you folks haven't had some severe flooding on the Illinois River. Before I leave flooding, though, just to give you a feel, this is information from the St. Louis gauge that goes all the way back to 1860, right before the Civil War started. And so this trend in the number of days the river's in flood stage has been increasing throughout that whole time period, as has the maximum elevation of the river. So this is a very long trend in this increase in flooding duration and magnitude. Some of that definitely is related to precipitation, but there's other things we've also done to this system that we shouldn't forget. Changes in the watershed and land use, impervious surfaces, lots of dams in the river, etc. All right, back to our analysis. So I used a mixed model repeated measures approach to look at these data. And when we throw everything in here, we've got year, the abundance of silver carp, the interaction between year and silver carp, and then our three covariates. Doing the analysis, only year, silver carp, and the interaction were significant. So we challenged them with those three covariates. None of them turned out to be significant, and silver carp were still a very significant effect. So really, in terms of using these sorts of data, this is very strong empirical evidence that there's a negative effect from silver carp on native sport fishes. I really can't think of how anyone is going to test this with any better data. You can also use that interaction effect, by the way, to say, well, when did silver carp really start having a negative effect on sport fishes? And that answer is right before 2004, which is kind of interesting. So they needed some time to get to a little higher level. However, very few adult sport fishes have strong dietary overlap with silver carp. So what is the mechanism? And I think most of us would already be thinking that mechanism is going to be in the early life stages, in the larval and juvenile stages of sport fishes that also consume zooplankton. So there could be direct competition with those life stages. So then I could just turn around that whole analysis, and instead of looking at adult sport fishes, stock size and larger, now looking at juvenile sport fishes, those that are less than stock size. When we run this model the exact same way, we get a very different result. Now only temperature falls out as a significant effect. And I will tell you that when I do the simple ANOVA approach to BACI, that was significant. So this is a support for why you do the more complicated analysis, because only temperature is falling out. So something else is going on different from what went on with the adults. I decided to look at a more simple model, and I just reduced it to silver carp, temperature, and their interaction. Now all of those effects are significant, especially the interaction. So what's going on is in the upper reaches where there's no silver carp, the abundance of the juvenile sport fishes increases with temperature. That's actually a common pattern in fish ecology. The idea is if it's warmer, you grow faster, bigger is better, you're able to survive in higher numbers. We actually found that with some of our fishes in Pool 26 when we analyzed data from 1994 through 2004. But when we look at all the data, there is no relationship between juvenile sport fishes and temperature in the lower reach. So this is consistent with a notion that has been put out called the biotic-abiotic constraining hypothesis by Quist and his colleagues, which basically says that in certain situations you can have favorable abiotic conditions for recruitment, but a biological effect can constrain that recruitment. And so this would suggest that silver carp may be constraining recruitment in these lower reaches. All right, summarizing, we've got strong empirical evidence of a negative effect of silver carp on adult sport fishes. The relationship between silver carp and juvenile sport fish was more complicated. It's an interaction with temperature. And basically, it's consistent with the notion of the biotic-abiotic constraining hypothesis that silver carp may be constraining sport fish recruitment where they're present. Now, we're going to get finally to the lighthearted, romantic portion of this this talk. So we're going to talk a little bit about silver carp reproduction, but really what I'm talking about is the behavior of the very small larval silver carp. And this information is coming from this NSF Research Experience for Undergraduate project that I was involved with. This was an interdisciplinary project where these students were coming in and studying both archaeology and ecology. We did split them up into groups. There were half the students that we selected that were primarily interested in archaeology and half that were primarily interested in ecology. And then they formed interdisciplinary groups, and we gave them long-term data from both archaeology and our long-term monitoring data from LTEF and LTRM, and they compared community structure between the archaeological data and the modern data. So interesting stuff. They're going to present those posters that they did up at the Mississippi River Research Consortium in April. The program began on June 15th, and it ended with a public poster presentation on August 7th. I just want you to remember those dates. It was a real great experience for me because I don't get to do a lot of teaching at a field station to work with these kids. Here they are, mostly the fish group out with my crew. And I think they had a blast and they learned a lot. They did great projects. But I wanted you to remember those dates because flooding, yes. So think of, look at where June 15th is on this graph. And here's where flood stage is, and here's where August 7th is. So the entire time they were trying to go out on this river and do their projects, there was moderate to major flooding going on in these rivers. And that made it very challenging for them to go out and do our routine monitoring because often we couldn't go out and do our routine monitoring. So we had to find things for them to do. And for a long time when there's a major flood, we've been seeing large schools of very small fish at the edge of the flood. And here's an example of that. So I think you can all see lots of little fish swimming there. And we've always wondered, well, I mean, they're very small. You can't identify them with your eye standing there, but a lot of times we're thinking those might be silver carp. So we took advantage of having all these kids and we went to these areas where floods were just starting to come over a road, just starting to go into a yard, things like that. And we wanted to sample these fish, but how do you sample these little tiny larval fish in about 2 inches of water? Luckily, I was able to get, we have a little slush fund that we hide from people, and we use that money. We invested about, really all in all, close to $500,000 and brought in some civil engineers to help us design a new monitoring device. And I'm going to unveil it for you right now. We were really lucky we had that money. Because this is what they came up with. Right? So actually this is what we did. We just duct taped this little aquarium net to poles and sent them out. But we tried to do this as, kind of a repeatable study. So they went to a bunch of different locations. At each one of those sites, we tried to have 3 people sampling. We timed them. They would dip for 5 minutes, and that's a sample. We sent them all the way south towards St. Louis, and then we went all the way up towards Lock and Dam 25 in Winfield. We sampled both the Missouri side of the river and the Illinois side of the river. And believe it or not, those little aquarium nets captured a lot of little larval fishes. That pan is 1 sample. In total, we captured 8 taxa. 95% of them were silver carp. We captured over 12,000 silver carp dipping with those little tiny aquarium nets. That really tells you their affinity for moving with the edge of the flood. Here's the length distribution of those fish. Basically, the mean was 10 millimeter standard length, varying from about 5 millimeters, a little yolk sac larvae there, up to 15 plus, up to about 21 millimeters standard length. So these are post yolk sac larvae that are just beginning to transition into juveniles. And looking at our catch per dip, I guess you would say, number of silver carp captured per sample, the confluence of the Illinois and Mississippi River was a tremendous hotspot. They were sampling on average 2,000 silver carp larvae in 5 minutes of dipping with that little aquarium net. just loading up. There was also fair numbers on the Illinois side below the confluence of the Illinois River. On the Missouri side, we had our greatest abundance above the Illinois River on the Missouri side. So, just a little bit of information there, but we had 8 taxa, over 12,700 individuals captured. Over 95% of them were those silver carp larvae. And really, this is telling me that we've really underappreciated the affinity, the behavior of these early larval silver carp to move at the edge of this flood and to get into these floodplain habitats. There's a lot we still need to learn about the ecology of those larval stages of silver carp. The confluence of the Mississippi and Illinois rivers was a hot zone. I wonder if that might happen in other confluences. My colleague Greg Whitledge at Southern Illinois University Carbondale has been doing a lot of interesting research where he looks at the isotopic signature of water, and then he looks at the isotopic signature of otoliths, and he can tell what part of the river those fish were spawned in and had their natal environment, basically. I'm wondering if we took isotopic signatures during these major floods, and at the edge of that flood, whether that isotopic signature might be different from non-flood times and from other locations. That could be some interesting work to look into in the future. So, just wrapping this up with some take-home messages. Thanks to the climate trends that we're seeing, geographic and population expansion of silver carp likely to continue in the upper Mississippi River system. We've still got much to learn about the larval and juvenile ecology of silver carp. Then when I think about our backwater lakes in the lower portion of our system are really plankton factories. We can't grow aquatic vegetation in them anymore because of all the sedimentation that's coming in. What should we do with these backwater lakes? Should we renew our efforts to reestablish aquatic vegetation into that habitat? That's going to be very challenging. It's going to cost a lot of money. We could isolate all of these backwater lakes, take them away from the river, keep silver carp from having great reproduction and foraging habitat there. Again, that's going to be very expensive, but very dramatic, but these may be ways we can reduce the populations of silver carp. And I wonder what the value of these backwater lakes are in their current form when they're contiguous with the river. So a lot we could think about moving forward. The last take-home message, really, the value of that long-term data cannot be overestimated. The things we can do with it now, with these 20 years, more than 20 years really, truly astounding. We keep learning that each year. I'm going to end with what about the Great Lakes? The bad news is the prey resource available for Asian carp in Lake Michigan, I believe, has been very much underestimated. And to learn more about that, please go see Sarah Creaky's presentation tomorrow at 11 o'clock, looking at the underestimation of microzooplankton in the Great Lakes. And then tonight at 5 o'clock, please go see Emily Reed's poster looking at the nearshore rotifer community in Lake Michigan. And you get a little better feel for what's available for Asian carp in Lake Michigan. If there's any ray of hope I can give you is thanks to some work that Ed Culver did as a graduate student. We know that silver carp are very vulnerable to injury from electric fields. In his study, which is out now in North American Journal of Fisheries, for example, on the Illinois River, we had 71% spinal injury rate to silver carp. And these injuries could be massive to their spines. We also learned in that study that pulse frequency is a key parameter that could be adjusted to increase the injury rate to silver carp. And compared to our native fishes, the ones that we tested had very low injury rates from electric fields. So this at least raises the possibility that maybe there's things we could do with those electric barriers to make them more effective by basically potentially injuring and killing silver carp, which does not raise any issues with me, and it shouldn't with you. In the end, I also want to thank all of my crew down at Pool 26. That's Eric Gittinger. We've been wondering what's happened to Ed Culver's brain capacity, and now we know. That's Lori Ginninger and Megan Cowan out doing water quality sampling. That's Ben Lubinsky, the most cheerful human being in the world. Eric Ratcliffe, the hungriest human being in the world. And then 3 of our hourly technicians. We've got Ryan, Eric the Third, as we call him, because we've got 2 other Erics already. And this is Chris Maxon. Chris will be giving another presentation on Wednesday. And that crew is really, in the history of the field station, that's the best crew I've had out there over the last couple of weeks. I do really appreciate what they do for me. And I'm going to end by saying I do have a graduate assistantship available for someone interested in pursuing a PhD. And if you're interested in that, come and see me sometime during this meeting. Thank you very much for your attention.
We've got time for questions.